everyone, my name is Jiyoon BAE from the KGSEA Math Circle, and today I'll walk you through the basics of gene regulation, focusing on the concepts of operons. And today I'll walk you through the basics of gene regulation, focusing on the concepts of operons. To begin with, gene regulation is the control of gene expression, and in other words, it is how cells manage when and how much of a gene's product is made. This means turning genes on and off or adjusting how active they are. But why would cells regulate genes in the first place? There are several reasons why. First of all, it's for cellular function. Producing proteins takes energy, so it is wasteful to express unnecessary genes in a changing environment. Second, it's for development and cell specialization. For instance, nerve cells and muscle cells have different functions, so they must express different genes. And third, to maintain cellular homeostasis or a stable internal environment. In prokaryotics, gene regulation needs to be especially quick and efficient. Since they lack a nucleus, unlike eukaryotic cells, their DNA is free in the cytoplasm, so transcription and translation occur simultaneously. As a result, they need fast ways to respond to changing environment. Regarding such issues, the main mechanism utilized is the operon system. So what is an operon? It is a cluster of genes that are all controlled by the promoter, which are eventually transcribed into one mRNA. Think of it like a power strip. You plug in several things, but there's just one switch to control them all. This system is common in bacteria like E. coli for its development through coordinated gene expression. Moving on, we'll cover the structure of an operon. So an operon has several key parts. First, promoter. This is where RNA polymerase attaches to the DNA. If RNA polymerase cannot bind here, the whole process does not even start. Next up, the operator is where a repressor protein binds to, and the binding blocks RNA polymerase from moving forward. That means that the structural genes won't be transcribed. So when the operator is blocked, the genes are off. When the operator is free, the genes are on. Structural genes are the genes that function to make the proteins like enzymes. As mentioned beforehand, these genes are grouped together and transcribed at once into a single mRNA molecule as shown here. After the RNA molecule transcribes the genes into mRNA, it will later be translated into final product or proteins, which will be done by ribosomes. Regulatory gene isn't technically inside the operon, but it plays an important role. The regulatory gene makes the repressor protein, which represses and turns off the operon. This gene is always being transcribed at a low level, so the repressor protein is always available to act when needed. To keep it short, promoter is where the RNA polymerase starts, operator is the on and off switch, structural genes are the actual genes that make proteins, and regulatory gene makes the repressor, which turns the operon off. There are two types of operons. First, inducible, and next, repressible. Inducible operons are usually off by default. That means the repressor protein is active and blocks transcription unless the operon turns on. Repressible operons, on the other hand, they are usually on by default due to its inactive repressor protein. The difference is quite straightforward. It depends on the default setting, whether the genes are normally being expressed or not. A well-known example of an inducible operon is the lac operon in E. coli. This operon allows bacteria to digest lactose, which is a sugar, but it only turns on in the presence of lactose. Otherwise, there's no point in making enzymes to break it down. So the lac operon breaks lactose into usable sugars, and it's part of a catabolic pathway. That means it helps the cell break down a molecule to release energy. By default, this operon is off. It only turns on when lactose is present. So now let's dive into the details. First, what happens when there's no lactose? The repressor protein is produced by the regulatory gene. This repressor binds the operator, which is between the promoter and the structural genes. Because of this, RNA polymerase is blocked. It can't reach the genes to start transcription. So no lactase enzyme is produced. This is because if there's no lactose to digest, there's no reason to make the enzymes that digest it. This is how operons save energy. Moving on, when the lactose is present, it binds the repressor protein. This changes the shape of the repressor, making it unable to bind to the operator. Without the repressor in the way, RNA polymerase can move forward from the promoter and transcribe the structural genes. These genes code for enzymes like lactase, which break down lactose. So here, lactose acts as the inducer. It deactivates the repressor and allows transcription to happen. Now let's cover the trip operon, which is a repressible operon. This one controls the synthesis of tryptophan, which is an essential amino acid. Tryptophan is important for bacterial growth and function. One significant difference from the inducible operon is that repressible operons go through an anabolic pathway. It builds tryptophan instead of breaking it down. When tryptophan levels are high, tryptophan molecules bind to the repressor protein, and this activates the repressor. The repressor binds the operator and blocks transcription, so the bacteria stop making more tryptophan. In this case, tryptophan is the co-repressor. It works along with the repressor protein to turn the operon off. However, when tryptophan levels are low, the repressor protein remains inactive on its own. It cannot bind to the operator, so RNA polymerase moves through and transcribes the genes that make the enzymes to synthesize tryptophan. An important note here, the regulatory gene is always active, so the repressor protein is always being made regardless of the level of tryptophan. But whether the operator is active or not, it depends on the binding with the repressor protein. Now we'll connect this to a bigger idea, which is feedback loops. A positive feedback loop means the output amplifies the process. For instance, in a concert, when fans cheer louder, the performer will give more energy, which eventually makes the fans go even louder. A negative feedback loop means the output reduces the process. Like when your body gets too hot, you sweat, which cools you down, and then you will stop sweating. These loops are really common in biology because they help systems stay balanced. So back again to the topic, how do these feedback loops apply to operons? Both the lac and trip operons are examples of negative feedback. In the lac operon, once lactose is digested, there's no more inducer. The repressor becomes active again, binds the operator, and stops transcription. In the trip operon, once enough tryptophan is made, it binds the repressor and the operon is shut down. So in both cases, the end product reduces further production, which helps the cell stay efficient and avoid wasting energy. So the key takeaways from today is that first, gene regulation is how cells control which genes are expressed, and operons are a system of grouped genes controlled by one promoter and an operator, commonly used in bacteria. The lac operon is inducible, which means it's off by default and turns on when lactose is present. On the other hand, the trip operon is repressible and is on by default and turns off when tryptophan is present. Both systems are based on negative feedback, helping the cell save energy and respond to its environment. This is it for today, and thank you so much for following along.